The webinar today is about quantitative analysis of 2D e-gels, and as George mentioned, why being based on warping, spot detection, and normalization, spot analysis, quantitative analysis is much more reliable. Let's just dive into the matter. Biological systems, are they deterministic systems? Or in other words, why are we doing all this stuff? Why do we want to quantify protein expression? If it would be deterministic systems, things would be rather easy. Imagine a certain parameter in an organism that you are studying would have always the same quantity. Then in any of our observations, any experiments, we would see the same quantity. So with increasing number of observations, we would just see the same quantity again. However, since biological systems are not deterministic, we don't have this nice kind of signal here. Instead, we always face bell-shaped distributed parameters. At least we expect or we assume bell-shaped distribution of these parameters. And these are typically represented by a certain mean and a certain standard deviation. The deviation is not only coming from the biological variation, it also comes from your technical system, the system which you are using to measure the system and the parameters in the system. For example, 2D electrophoresis, including taking samples, preparing the samples, labeling them, and so on. So a lot of technical steps are involved, introducing additional variation. And this is the reason for doing quantitative analysis. We want to have reliable quantities, we want to have them reproducible and have good and interesting results. How can we deal with this variation? Professor Meyer, coming from Bochum, once told us that the most common solution in the world is that experiments are performed only once. We don't run replicates, we don't have certain variations if we just have one value for each parameter. Then, of course, you can bring the biological variance to zero if you pool your samples. Just pool all your different samples together in one sample and just analyze this one. Or make technical replicates from the sample and call them independent. Just define them as being independent. Rename qualitative data as quantitative, or use improved statistical methods which bring what you want to see. Of course, all this is just kidding, and the less common solution is to really work scientifically, and that's what we, of course, recommend and what you all want to do. And for this reason, I want to give some hints and introduction how to do this and how to do this with delta 2D. What we are interested in is complete and interesting expression profiles. So the basis for quantitative analysis is to have complete expression profiles. Complete means we have a certain protein quantity across all the different gels and images that we have. This is rather simple if we have multiple images from the same gel, because then we have just one gel without any distortions. There are no variations in the positions of the spots among the different images. However, it becomes more complex if we have multiple images from multiple gels, introducing the distortions between the gels. How can we get interesting expression profiles even then? For the first problem, the distortions, we want to warp the images. We want to align them and get rid of different positions of protein spots across the gels. In delta 2D, this is the step 2 of our recommended workflow. Step 1 is setting up the project. We have had 2 webinars about this in the past, one from Tomasz Burszikowski and also another one from me. Tomasz from the University of Hasselt has been talking about principles of experimental design, and I then have been talking about how to do this and set up projects in delta 2D. And now, based on that, we want to warp images along their similarity. How can that be done? Or what is warping? as a basis, imagine you have a certain gel image, and then you have a second one, and you put them on top of each other. One is colored in blue, the other in orange, and then you can see there might be some similarities, but it's really hard to identify which spots belongs to which other one and which is the stronger one on which of the two images. Warping means we eliminate the distortions, and then using this coloring, which means the black shades of the colors of the spots belong to the same spot and have an overlay now of the same intensity. Different shades of gray mean that the spots have the same intensity but are not really very strong spots on both images, while those spots which are present on one or the other image only, or very much more intensive than on the other one, are blue or orange, respectively, on which gel they are more intensive. So the image warping leads to perfect gels. It compensates for the running differences. It's fast and reliable, and it allows for the visual control and at least qualitative analysis. And we have introduced this 13 years ago with our first version of Delta 2D. Now, coming to more than two gels, this becomes a little more complex, and you could now imagine that you have to choose one gel as the reference gel for all the other ones. If you have an experiment of, for example, 6 gels, one solution could be to warp and align all to one reference gel. We thought this is not the best idea. It should be something like more flexible, and you should be able to warp along similarities. And replicates should be very similar by definition, because they should belong the same information from the same sample. That's why we said we should be able to warp between replicates within replicate groups, which would be, for example, connections like this and this and this. And then we minimize the number of alignments between different replicates. And for example, we would here take the first of the second group and warp it to the first here, and first of the third group and warp it to the first of the first group. And with this, we have what we call a warp graph. Every image is connected to every other image, and Delta 2D can combine all these transformations so that we can easily compare the second of the first group with the second of the third group and get access to a visual comparison of these two gels. This approach saves time. You don't have to check each possible combination or image pair. That means you just have to check and warp n-1 pairs for n gels. For 20, you have just 19 warpings, and not every possible pairwise combination needs to be warped directly. And it improves the quality because we can warp along the similarities as I have explained. So you are very free to define such kind of warp graphs. Let me show this in Delta 2D. Let's switch to Delta 2D and a nice experiment that we want to use. This is a sample experiment that comes with the installation of Delta 2D. We have 3 different groups, and these groups belong or contain 2 replicates each. One is the control, one is taken after 1 minute sample, another sample is taken after 10 minutes. Now, this is done in the first step, warping project setup. Now we want to have a look at the warping setup. And one very flexible and manual way to warp images or define the way how to warp this experiment is to just drag and drop the images on top of each other. So I can now just take the second replicate of the 1 minute group and drop it on the first replicate and let Delta 2D know these 2 images should be warped to each other. I can do the same for all the other groups. This is done quickly and rather easy. Now they are connected already, and well, I have to admit that there is a preparation done. There are already warpings behind available. That's why these bullets are green already. So now what we still miss is connections between the different replicate groups. Let us do this that way, that I just take the first replicate of the 10 minutes group and drop it to the first replicate of the control group, like this, and also the first replicate of the 1 minute group, and warp it also to the first replicate of the control group, like this. Now we have connected all of them, and there is no image which is not yet connected to our graph and not bound into this warp graph. Of course, we can also apply strategies instead of doing that manually. For large projects, that would be very time consuming and you might get lost. So the first step is then, again, defining all the groups, and you should then define the best image of each group as the first one. You can easily change the order of these two images within each group. And if you think you should change that, because the best should be the first, you can easily do that, like this. And then you can apply in the warping setup a strategy. You can right click here and choose Set Warp Strategy. Or in the workflow, it is offered in the second step, Warp Images, to define a warp strategy. A dialog will open which provides 3 different strategies. If you are running a Deutsch project or 4, and you will have an additional one for using internal standards. In this case, we would always recommend group warping strategy. The group warping strategy means what I have described already. Within each replicate group, the images are, or the gels are warped to the best, the first one, and between the groups, the best of each group is warped to the best of the first group. Then you can define whether Delta2D should try this automatically or whether you want to do it manually with different warp modes. I press OK, and then this strategy would be applied. Let me first delete all these connections. I just click them and can delete them pressing the delete key. I can also right click and use the context menu to choose delete and delete these connections, like this. And then I choose warp strategy again, choose this group warping strategy instead of, for example, all to one, where I would have to choose one as the reference image, or a chained one if there are time points, different time points, and it would be easy to connect them with neighbored points in time. We recommend the group warping strategy, as I said, and if I apply that, all these images would be connected automatically. And this indeed makes sense for large projects. Then coming to the different warpings themselves, we should then open a certain transformation which is not yet done, at least if you are starting with that. I would first have to delete all these match vectors because they are not yet available. Now we have the raw images. They are there. Let me increase the size of this image and zoom out a little bit. Now this is an overlay of the 2 replicate images from the 1 minute group. And as you can see, there are a lot of spots on both images, and it's rather difficult to find patterns that are available on both. Of course, there are some here. For example, there's a set of spots which appear on both. And you cannot just move one image so that the spot positions are the same. We really have to warp and align images in a sophisticated way. Now, since the match vectors are deleted, you can now see in this warp setup this first this bullet here, and it would even change since we recommend also to let the automatic warp mode give a try, and then this becomes such a symbol like this. That means there is a connection defined. It's not yet calculated, but Delta2D can do this automatically and at least guess some matches and match vectors. Let's have a look how this is done. I just press the warp button, and then Delta2D is analyzing the images. The complete image is taken into consideration, and then match vectors connecting similar areas on both images will be found. We will soon have a look at the result. This is image processing. It takes a second depending upon the power of your processor and the memory, of course, which is available in your machine. So this is almost ready. Now we have that. As you can see, there's a number of match vectors. There's a number given in the status line. Delta T has found 36 match vectors, and we can now zoom in and review this if we like, and have a look at the result of this warping. And as you can see, which is typical, close to the borders, there are still some shifts in spot positions, which you can then correct. And you can do this stepwise. We never said warping will be done automatically for all possible images, but I think, as you can see here, it's done for a large area. It's ready, it's fine with that. And we just have to improve it at the corners at some situations where Delta D couldn't find proper matches. How to improve that? We can now use this position as the starting position for another automatic warping. If I now say automatic and let Delta D warp again, it would take the existing match vectors. Of course, we could delete those match vectors which have been found by error. Also, this can happen. And we can also, instead of doing and applying automatic warping again, we can add match vectors manually, for example, by clicking on a certain position on an orange spot and then on a respective blue spot and press warp to apply this match vector. Defining match vectors can be easy if you use a snap to spot feature. Then the end of match vectors will be automatically snapped into the next existing spot maximum. We can activate this in the options. There is this parameter snap match vectors to spots. I activate that, press OK, and then next time let's have a look for additional pairs that need to be warped. We have this in this upper left region, and then I just click next to this orange spot and next to the blue, and as you can see, hopefully the match vector is automatically snapped to the respective two spots, and then it's done rather easy to define additional match vectors. And we apply them, and as you can see, it's done really quickly to improve the matching and the warping which is available on for this image pair here at least. There could be some fine-tuning also be necessary across the images, and of course we recommend to do this very careful to really review the warping, because everything else will then later be based upon this image warping. And the better it is, the better your results will be, of course. So review the warpings for all your image pairs carefully, improve it if necessary, and then once you are ready, you can approve all them, and in the workflow panel you can see that this warping is now done. The wheel has changed to this other orange symbol, which means Delta Tree has found automatic match vectors. You should review that, and once you are ready with reviewing this pair, you click just approve, and then all match vectors are accepted by Delta 2D, and then you can see this green symbol. And you should do this for all the existing pairs. As you could see in the workflow panel, there is a list of all the possible pairs in your project, and when you're ready, all of them should be green as they are now. So this is what I wanted to explain with respect to warping. If there are questions about that, you can ask them in the chat room. Feel invited to ask any question. We will try to answer them after the webinar, or send us an email. Then, based on the image warping, we come to the next step, which is detect and quantify spots. This is also available in the workflow panel. It's the next step. It's really called detect and quantify spots, and it guides you through what should be done next. The first is fuse all images. Image fusion is something very useful. We have introduced this to create one image which includes all the spots. As you can see now, if you fuse the image, we get a resulting image where all the spots are collected from all the original images, and from whichever image they come, they appear together here, even if the proteins would never appear at the same time in the organism you study. So this could also serve then as a proteome map for presentations where you show all your proteins in one view and you don't have to point to spots on different images to really explain the proteins that you have found in your experiment. This is unique in Delta 2D. We have invented this in 2003, and this helps a lot to get advanced on the way to complete expression profiles. At the same time, when doing this, a lot of things are done with the images. One is in fact done when you import images. Speckles will be removed if you like, and also background is removed when fusing the images so that the result, the fused image, will have a clear contrast and the background is not visible anymore. It does not disturb your analysis anymore. You can at any point of time review the background which has been subtracted or the cleaned image to see that no spots have been subtracted along with the background and that there is almost no background left on the cleaned image. And then we want to identify objects on the fused image. This is done by spot detection, and of course, to do this, we have some helpful views and changes in the settings of the views. For example, you can adjust the histogram settings. With one of the last versions of Delta 2D, we have introduced that the contrast is set automatically during the import of images, and we also offer a gel image regions view where you can see the same region across all the images. Of course, the histogram settings do not affect the quantities. Quantitation is done on the original images, of course, after background has been subtracted. But the contrast settings do not affect quantitation of spots. It just affects the display of the images. Regarding the spot detection itself, there are a lot of parameters or some parameters available. They are chosen automatically from the image. The images are, or the fused image is analyzed, and then Delta 2D is providing some guesses for spot detection for the parameters, and the default settings are rather sensitive because we think it's much easier to eliminate false positive spots from a spot detection rather than to add additional spots. Background subtraction is done by a certain parameter. It's controlled by this parameter called local background region. There is kind of a parameter for noise filter. It's called average spot size. There is a parameter for discriminating small and large spots. What is a spot? From which spot intensity do I want to consider a signal to be a spot? It's called sensitivity in percent. And we also have a nice parameter which is controlling the shape of the spots. If you have seen spot detections done by really basic spot detection algorithms, then they look rather, unpretty. They are not very nicely shaped. And what we can do is we model the spot shapes so that they have nice shapes, which is improving also the quantities of the spots, and it's increasing the reproducibility of quantities, as we could show in one publication years ago. After doing spot detection, I will show this in a minute in Delta 2D, you can improve this because we, again, as for the warping, we never claim for having optimal algorithms. We think that they are not available actually. There is no chance to get a really optimal warping for images, and there is no chance to really get an optimal spot detection for any kind of different image qualities. That's why we also allow for spot editing, where you can define this or change the spot shapes. These spot shapes always define the area which is quantified for a spot. So you can remove these shapes if you have the feeling that there are weak spots detected. As I mentioned, the spot detection is rather sensitive, so that you can get rid of very small signals. And then you can also split spots if they have been detected together by error, or you can add spots if Delta 2D has missed a spot on a certain area, and you can merge spots if you want to sum up the quantities of two neighbored spots. This means you optimize the spot pattern, and it is done just once on the fused image, and it doesn't matter which size your project has, because you just have one fused image where you do the spot detection and also the spot editing. And this means you will face significant time savings compared to the situation where you have spot detections across all the different images. And by the way, this is the second solution for getting complete expression profiles. Imagine you would have two images and do the spot detection on the two images separately. Then you might have 450 spots detected on one image and 560 on the others. And that doesn't mean that the 450 from the one image are included in the 560 of the others. You will have asynchronous spot detection, having missing partners on both images. And this is rather difficult then to guess which spot on the other image belongs to this one on the one image. And for this reason, we thought it is good to have this fused image, which serves as a template then for all the other images. And what we do is we see a signal on the fused image. We know there is a signal on at least one image. And then we want to have this signal measured across all the different images. That's the idea behind, and for this reason, we will get complete expression profiles having a partner spot for each spot across all the images. This is done then by transferring the spot boundaries from the fused image to all the other original images. So we have detected and edited the spots to get a real reliable spot pattern, a consensus spot pattern, and then we transfer it to all the original images, and then we have the same number of spots across all images, and we have the same pattern, and we have a complete spot matching, and all this is done automatically by Delta 2D, and this is then added to the warping and providing 100% complete expression profiles. This is a unique feature in Delta 2D since 10 years now. No other software package has been copying this behavior. Let me show this in Delta 2D. In Delta 2D, we have seen we have seen in the light table already that these images are grouped. We have warped them, and assuming that the warping is fine, we can now fuse them. Let me click on the fuse images button, or let's choose the workflow and switch to step 3, where we have access to fuse all images feature, and this is now opening the dialog where we want to choose the images which we need to fuse. If you have a large number of replicates and know that the replicates are very well reproduced, then you can exclude certain images from the fusion, because having the same image twice doesn't help or doesn't improve the quality of the fused image. So the idea behind is to fuse as many images as necessary, but as few as possible to get an image where you collect all the spots on it. So if you are not sure whether control 2 has additional spots compared to control 1, then include it in the fusion. If you know control 1 includes all the spots from the sample and control 2 does not add additional spots to it, then you can exclude the second image from the image fusion. There are some filters available. You can remove the background of the original images before fusing them. That's what we would recommend. Amplitude rescale is increasing the contrast, and we want to use union fusion. And then also you have to define a certain master gel image, which is then the reference gel with respect to the spot positions. So all the other images will be warped to this one and then fused to a fused image. I now press fuse, and then Delta2D is analyzing all my 6 images, warping them to the control 1, and then joining all these images, adding a fused image to my panel, to my warp graph. It's now attached to my control 1 with a certain warp relation. And we can now also open it either by double-clicking this image, or in the workflow it's available to directly detect the spots on this fused image. Let me first open it so that you have an impression of how the fused image is looking like. As you can see here, it's looking like a real gel image. You really can identify the spots that you typically know from other gels. You know which spot contains which proteins. It's your experience with your organism, and the fused image is keeping all this nice behavior or nice impression of your gels. It's a really nice looking gel image where background has been subtracted. So let's now detect spots on this image. I click the detect spots command in the workflow on the fused image. The spot detection parameters are described in this part of the dialog. If you click into the parameter field, the description is shown here and you can read it and will change all these parameters. By the way, you can create as many fused images as you like with different settings, maybe change the warping, improve the warping, do a fusion again. It doesn't matter, but then of course you have to be aware on which image you want to detect the spots, which fused image you want to use as the basis for the spot transfer later on. So don't get lost in all the different fused images. And on a certain fused image, as we have it here, on this fused image, we can repeat the spot detection as often as we like. So I can now play with these parameters, change them, and see how changes translate into different spot patterns. If I then later want to come back, and let me, for example, change this to 50 so that I don't get very small spots, just big ones. And if I then later think, oh, this hasn't been a good choice, I would like to get back to the original parameters as they have been guessed and estimated and proposed by Delta2D, then you can just click here this button and then you get back the original parameters that Delta2D has taken from this image. Let's try the spot detection with these parameters. I press OK, and then the spot detection is done on my fused image only. As you can see here, there is a number of spots detected. 1,102 spots have been detected. And of course, the image includes areas where we don't want to use the spots. Of course, Delta2D does not know which area is of interest and which not. So we can remove those spots from areas that we are not interested in now manually. I can just drag a rectangle with the activated spot selection tool here. I can just choose a certain rectangle, select all the spots, and then press the delete key on my keyboard or right click on one of them and press cancel spots here. And then they are hidden automatically and ignored by the further analysis. Furthermore, we can zoom in and have a look at the spots whether they are detected properly. For this, I could choose the zoom tool now, or keep the spot selection tool activated and choose the control key and my mouse wheel to zoom in to a certain area where I'm pointing to. And what we recommend is to really check this spot pattern very carefully. You do this just once on one fused image, typically, and for this reason, it might take up to an hour or even 1.5 or 2. Be careful and improve it, optimize it, so that you are really satisfied with the quality of this pattern. This is the pattern that will be applied to all the other images later on. So for example, we would maybe delete very weak spots, maybe like this one or this one or this one. I can now choose them and select them one by one and press the delete key to get rid of them. I could also open a certain table to select for a certain spot quality. Maybe we can do that. In the project explorer, we have the fused image also added to this project, and we can click it and open a certain quantitation tool only including this image. I now want to see the spot quality column as well, sort for this spot quality column, and then those spots which are very untypical. They do not have a typical bell-shaped distribution if you cut them in pieces, and then this is of very poor quality, while the others on the other end of the table, I sort it the other way now. They should have a very nice shape and they look very typical like a real spot. So starting with these very poor spots, I could now check on my dual view and on my fused image, is this a spot or not? And for example, this spot, I think, is very weak. It shouldn't be really considered. And in this way, I can now choose, sorry, this is not the right table. This is the table for the fused image. I now can choose better and better qualities until I get a benchmark where I think from this quality on, I assume the spots to be spots and I want to keep them. So maybe in this table, we get up to qualities of this. This is still a very weak spot. I wouldn't consider this to be a spot. We can, of course, use a roll up, like the 3D spot roll up, and then check the spot whether it's a signal or not. It's a very flat signal here, so I wouldn't consider this to be a spot. So let's look for even larger spot qualities, like this. And once I see, okay, now I would say this is a spot. It's starting with this spot quality. I would like to eliminate and get rid of all those with less spot quality. I click one of them and go to the top of the table and shift click the first one to select the whole region of this spot quality to my benchmark, and then I just can say cancel the selected spots. And now a lot of spots have been deleted from my fused image. So this is a quick way to get rid of weak spots, but that's not the whole story. The spot detection, as I mentioned, can also detect spots together which should be separated. For example, like here, these spots should be split into pieces to quantify the different spots separately. For this, I have to activate the spot editing tool. And then I can just click on the second spot here and maybe also on this one there to get and to separate the spots with respect to my expectations. So you just click on where you want to have an additional spot, and then Delta 2D will automatically define the spot boundaries using the same algorithms as always, and that is what we think is the better way, rather than to let you paint spot boundaries and cut them with a knife into pieces, because everyone would do this in rather different ways, and this would of course affect the spot quantities. So Delta 2D should do this with using the same algorithms as always. Again, here we should split this spot into pieces, also in this region, and you should systematically scroll through your image, starting maybe with this upper left corner, adding spots where you want to add spots, for example, here, here. And I don't want to do this very careful because this would take maybe some time, and I just want to show the principle how to do it. Here maybe also here, and here, and here. And then you scroll to the right, improve the pattern there, scroll down a little bit, check the pattern here, like this, scroll back to the left, scroll down again, and then to the right again. And once you have scrolled through the complete image, you could say, now I'm done. The spot pattern is looking like it should be, and then you can transfer that. In the workflow panel, Delta2D has now recognized that the fused image has spots, and that's why we could now open the fused image. Of course, this is done already. And then once we are ready, we can transfer the spots from the fused image to all the other images. And this is of course done with respect to the warpings, and then we transfer these spots to the other original images. This is done already. And once we have done this, we can now also, of course, we can at any point in time have a look at the gel image regions, where we see the same region across all the images. And if you are in doubt whether a spot on the fused image is 1 or 2, you can also use this gel image regions view. For example, here we have a nice spot on the fused image. Let's use the histogram settings to increase this and to remove the background. And then we can see that, in fact, this spot should be split into 2 because it appears twice, or 2 spots appear on all the images. That's why we should split this spot into on the fused image. This is a good decision, and the same applies maybe for this spot here. Let's have a look there. Oh yes, indeed. So the gel image regions view provides a nice possibility to review a certain area across all the images, and then again improve the spot pattern on the fused image. However, this pattern is not automatically used on all the other images. Once you are ready again, you have to do the transfer of the spots again. Let me have a look also on this spot here. As we can see, there is a weak signal next to this and strong here. I should also improve this situation here on the fused image, and also for this spot very close to it. So again, once this is ready, I choose again transfer spots to transfer the new and improved spot pattern to the other images. And as you can see, the new pattern is popping up across all the images now in this gel image regions view as well. Yeah, then at the same time, when we transfer the spots to all the other images, the spot boundaries are also remodeled on the target images. That means the spot boundaries do not look the same across all the images. They are adapted to the real images. And once we have all these spot boundaries across all the images, the spot quantities are measured. So this is done automatically in the background, and also normalization is done. So now what is normalization and why is it necessary? Imagine you have these 6 images here, and you can see that the first, maybe 4, have a less intensity compared to the last 2 images. That means the last 2 might have been stained longer, or the exposure time in the camera or system or on the scanner, the intensity of the light source might have been stronger. So they have just been measured more intensive. That doesn't mean that the protein expression is higher in general. The theory or the hypothesis is that a certain cell or organism always has the same energy available for producing protein, so that the whole amount of protein is always somehow constant. So normalization is done for eliminating these different levels of intensities between the different images. And normalization is done by summing up all the spots on a certain image and then dividing a certain normalized, a certain spot volume by the sum of all spots. And this is then resulting in the normalized spot volume of a certain spot. So normalization is done within each single image. And since if you consider this, the quantities collected in a certain table where every column represents a certain image, we call this vertical normalization. Within a certain column, the sum of the column is taken and each cell is divided by this sum. So that means the sum of all protein quantities, spot quantities, normalized spot quantities is then 1. There are some options, since sometimes you wouldn't like to have that all spots are 100%. You would like to maybe just choose a certain subset to be 100%. That means the sum that you are taking to divide all spots by it does not contain all spots, but a selected set of spots. Maybe just spot number 12, and then other spots, and spot number 245 again, and so on. So not just 1 to n, but selected spots. And there are two options. Maybe you just want to include spots where you know that they do not change in your experiment, housekeeping proteins or proteins that you have spiked into your samples, marker proteins. Or it could be that you want to exclude varying strong spots and keep all the others in the normalization basis set. And excluding or including is done in delta T by controlling a certain column where you can remove the check from this box and then you exclude this spot from the normalization basis. Remove it from the set of spots. Of course, these spots are also then normalized themselves, but they are not normalized by the whole set of spots, but just those spots that still have this checkbox activated. We have a small experiment prepared to show the effect of that. Imagine you have some synthetical spots, and all the spots have the same size across all the images, but just one spot, which is the central spot here, has an increasing amount of protein. Let me show this in delta 2D. Just a second. I would like to open this other project. I have to change the pool. I start these changes here. And I want to open the project which shows this example. Here we go. In the light table, we have all these different images, artificial images. And then let me show the quantitation table for it. We can now see, if we sort them, there are the unchanged spots which have the same spot quantity across all the images, but one spot is a growing spot. It has a small and then an increasing number of protein amount. And if we then have a look at this spot, we can of course also review it here. This is an example of the spots that do not change, and this is the spot that is increasing its volume. Well, now the normalized volumes do not change here because we already have excluded this one from the normalization basis. We only normalize all these spots by the sum of these spots, but excluding the spot that is changing. If we now include this spot, which is changing in our normalization basis, then in the normalization step, the basis for the normalization is changing because this spot volume is changing, and if it is included in the sum of all the spots, then this factor is changing. Let me do this. Normalization, include this spot in the normalization set. Now you can see that all these checkboxes are checked again, and then you can see that all the other spot quantities are going down since they are normalized against an increasing sum of spots. We can see this even better in the expression profile window. Here we have the growing spot and one of the other spots. It's unchanged, but in our measurement where we do not exclude this growing spot from the normalization basis, it would look like as if this spot would have a decreasing spot volume. That's why we want to exclude this large growing spot from our normalization basis, and you should do this typically if there are some few spots that are very large and are changing. So let me exclude this again, and then we come back to our normal spot volumes, and let me show this here. Just a second. It doesn't now update this view. Okay, here we go. Now this unchanged spot is unchanged again, and the growing spot shows this growing spot quantity. An additional step is done if you have Deutsch projects using an internal standard. Then every normalized spot volume is divided by the normalized spot volume of the respective spot on the standard image, on the site 2 image, or the G100 image if you are using the refraction 2D dies, and is then, multiplied by 100%, and then this is the spot volume that you get in delta 2D for a certain spot on a certain image. There are no options to control this step. This is just done automatically in the background when defining an experiment to be an experiment using an internal standard, and when you have assigned all images properly to certain gels and defined certain gel images as the standard images. The quantitative data is then shown in a quantitation table. There are different types of tables, all gel images and statistics tables. All gel images tables show single image values. Statistics tables only show group values, replication groups. There's a variety of attributes shown in different columns. You can sort by the columns. All tables can be sorted. Each row contains an expression profile, and of course you can sort and filter also the rows by the attributes. There's a visibility control so that you do not always see all these different attributes. You can change the visibility so that you just see what you want to see. And you also can create individual tables so that you do not get a certain big table for all the images or for all groups, but you can customize what you want to see. The visibility control is done by a certain dialog, or you can do this one by one in a certain table view itself. Let me show this then in delta 2D, because then we will see that there are complete and also interesting expression profiles, hopefully, but the interesting ones would be then found by using statistical analysis, which is part of another webinar later. Let me show this in delta 2D. The quantitation table is shown here. Let me double click to increase the size, but let me switch to the demo project again, where we have more data available. Let's go into the demo pool. Open demo project again. Having this standard quantitation table here, where we have the statistics view and also the all gel images view. You can control the visibility by clicking this eye, where you can see the different images, and then you can hide complete images from your tables, or you can hide just different attributes, for example, the labels, or show all the labels across all the images. You can hide or show ratios. You can have advanced attributes like the absolute spot volume in terms of gray values, the background that has been subtracted, or the spot area, for example. So there's a variety of attributes also for the statistics tables. And what we are typically interested in is, in fact, in the statistics, standard deviation, ratio, and the p-value with respect to students' t-tests. Click on the head of this column to sort column by this attribute. You can also invoke filters to then find interesting expression profiles. You can click a certain line, a certain expression profile, and then see this spot across all the different images and review whether this is a real spot signal or whether you should maybe exclude this from your further analysis. Like this. How to get customized table views. In this case, we have as a first group the control group, and then the 1 minute, and then the 10 minutes group. But maybe you want to also see the p-value comparing the 10 minutes group to the 1 minute group. Now the first group is the reference for ratio and p-value when comparing the groups with each other. You can now open the Project Explorer and then click on the 1 minute group and then pressing the Control key while clicking the 10 minutes group, and then pressing the quantitation table symbol will open a statistics table for these two groups, and then the first group that I have selected is the reference group, and the second is then showing ratio and p-value with respect to this first group. And if you have multiple groups, maybe 10 or 20 groups, you can select as many groups as you like, and they will show up in this table in the order that you have selected them. You can also show single images tables if you want to compare this 10-minute image with this 1-minute image, and then press the quantitation table again, the symbol for the quantitation table. You will get a table for these single image values, and the rule for what kind of table you will get, whether a statistics table with group values or with values for single images, is that if you at least click on one image rather than a group, you will get the single image table. If you just select groups, you will get a statistics table. Yeah, and changing the visibility, as I mentioned, you can choose this eye symbol to control the visibility in a very easy way, or you just click on columns in any table. You can do that to then check or uncheck certain columns for the different images that are involved in this view. Tables always allow for exporting them to Excel or to a character separated value format. You can also export pick lists, of course. Yeah, this is what I wanted to show in Delta 2D. Our approach with these two steps, warping images and then fusing images and doing the spot detection on just one fused image, transferring the spots then, resulting in complete expression profiles, saves a lot of time, and it is increasing the reliability a lot because you don't have missing values. And a remodeling of spots is done because this is also increasing your confidence into what you get. And the quantitation is always done on original unwarped images. We can always go back to the images as they come from your imaging device. The next steps are then part of another webinar. Join our next webinar, which should be held on September 10. It will be about statistical analysis, detect outliers and identify structures in your data, and of course find interesting spots. This is done with a sophisticated statistical toolbox as included in Delta 2D. And then let me also invite you to send us your publications so that we can include them on our reference list. let me end this webinar now. Thanks for your attention.